Welcome to Infectious Disease Lesson 2.2. What are the patterns of infectious disease transmission? The goals of this lesson are to analyze patterns of transmission to track down the source of an infectious disease outbreak, appreciate the importance of microscopy and cell culture for identifying microbes that cause infectious diseases, and to illustrate the concept of germ theory, that microbes arise from other microbes. At the end of the lesson, the students should be able to explain why it was hard to identify the source of an infectious disease before people knew they were caused by microbes, explain where microbes come from, and explain why viruses can pass through filters. We will achieve these goals by analyzing disease transmission patterns in the ghost map activity, and engaging in a Socratic discussion about historic discoveries that paved the way for the identification of infectious microbes. To prepare for this lesson, you'll need to review the following key scientific concepts. Transmission patterns can be used to identify the source of an infectious disease. Microbes are extraordinarily small and can only be observed under a microscope. Cell culture was vital for proving germ theory, and viruses are too small to see using a traditional microscope. You can review this scientific content in the background reading provided for you in the teacher primer, the teacher manual, and the student workbook. The teacher primer provides in-depth knowledge about the scientific content presented in this lesson. The teacher manual, or lesson plan, provides a minute-by-minute explanation of the lesson structure, and the student workbook provides additional explanation for students. Note that an annotated version is also available for teachers. Be sure to print the in-class worksheet, which is located in the Lesson 2.2 Curriculum Materials folder. The key points of the do now are that people once believed infectious disease resulted from poor morals or pollution. But today we know that infectious diseases come from microbes. But where do microbes come from? The do now begins by brainstorming where infectious microbes come from. Before microbes were discovered, people believed that infectious disease was caused by poor morals or bad air. Today we know that infectious microbes come from other microbes. This is called germ theory. The key point of the activity is that transmission patterns can be used to identify the source of an infectious disease. We'll get there by mapping a cholera outbreak in the ghost map activity. The ghost map activity begins by having students read about a cholera outbreak in London in 1854. The students then use the map and the worksheet questions to identify patterns of disease and to hypothesize potential causes for the outbreak. The key point of the discussion is that technology is required to identify microbes. Microscopes are used to see microbes. Culturing techniques can grow some microbes. Viruses are too small to see using a traditional microscope, and filters can separate viruses from bacteria. When discussing these historical discoveries, it is important to emphasize that microbes are still hard to identify today. Microbes are too small to see with the naked eye. It was only when the microscope was first invented that the new world of microbes was discovered, paving the way for the identification of microbes that cause disease. One major limitation of using microscopes to identify microbes that cause infectious disease is that it is hard to distinguish between them without using specialized staining techniques. A second important invention for studying infectious diseases was cell culture techniques. The ability to grow bacteria in culture also allowed us to prove germ theory, that is, that microbes don't spontaneously generate, they reproduce. Culture also allowed us to separate mixtures of bacteria into pure specimens, helping to prove that a particular bacterium causes a specific disease. One major limitation to using culturing techniques to find microbes that cause infectious disease is that we still cannot grow most microbes in culture. In fact, we think that even now we can only culture about 2% of the microbes found in nature. Viruses were first discovered when a filtering process was used to trap bacteria present in a liquid. The surprise came when the material that passed through the filter were able to infect tobacco plants. Because the bacteria had been trapped by the pores of the filter and weren't visible in the liquid that passed through, a smaller infectious agent, then called a virus, had to be present. But these viruses also couldn't be actually seen until a much stronger microscope, the electron microscope, had been invented. For a more detailed description of these discoveries, please refer to the teacher primer or teacher manual. The key points of the wrap-up are that microscopes and culturing techniques can be used to identify microbes. And we are covered in microbes, so it's hard to separate friend from foe. We'll get there by discussing how we might go about finding a microbe that caused a specific disease, and how we might distinguish friendly microbes from pathogenic microbes. The key point of the homework is that bacteria are everywhere, and we'll get there by having students write a paragraph about a bacterium of their choice that grow in extreme conditions. One of the most common misconceptions students have is thinking that we can grow any type of bacteria in the lab. Actually, only a very small percent of known bacteria can be cultured. We estimate about 2%. Another misconception students have is thinking that viruses can be seen under a microscope. Viruses are too small to see with a traditional microscope and can only be seen using an electron microscope. At the end of the lesson, collect the lesson worksheets to assess students' ability to identify patterns of disease and to hypothesize causes of outbreaks. This lesson primes students to think about some of the challenges we face when identifying the cause of infectious disease, which we will study in the next lessons. Don't forget, if you have any questions, concerns, or feedback, to let us know. You can contact any of the CTSE team members and we'll be happy to help you.